My experience with this. So I had influenza A, and it was the sickest I've been in many years. At one point, I had 102 fever. I denatured those viral proteins because I tried not to take anything for it. But once it hit 102, it's like I was worried it was going to get worse, and I needed to sleep. So I did take, I think I took one Tylenol for it at that point to try and bring it back down. But it's interesting because, and I think I said this on the Screencastify, it is important to get your vaccinations, even when, um, one of the things that the doctor told me when I went in was, you're vaccinated, so it would have been a lot worse than this if you hadn't been vaccinated. I mean, initially she was saying you'll probably be out 5 to 7 days. And I got it Sunday evening, late Sunday evening, and I'm back. And I'm not 100%. I'm still tired, but I'm fever free. So it is important to get those vaccinations, not just for you, but for others around you, because there's others people around you that you might not know are immunocompromised for some reason. There's a lot of people who are, actually, for all sorts. There's so many compromising diseases, so many diseases where your immune system is fighting you. And so people like that can go on autoimmune or can go on immunosuppressors to tamp down their immune system, which is great for whatever they happen to have. For example, I have rheumatoid arthritis. It's an autoimmune disease where my body attacks its own joints. I'm not currently medicated for it because I don't want to be immunocompromised. But there are a lot of people who are on immunocompromising drugs for things like RA. Diabetes type 1 is an is an immunocompromising disease as well. There's a ton of them. And so you getting your immunizations helps not only you recover quicker, but it also helps other people from getting it in the first place. And that's something to be aware of. So I recently just found out that someone I know pretty well chose, after getting their first COVID vaccination, chose to not get any more after the very first one. This is an educated person with a law degree. Decided not to get any more because of how they felt the first time. Remember, those are symptoms that are just there because your body thinks you're sick. You're not sick, but it's your body producing those antibodies that then will be stored so that when you actually get the disease or get an infection, you're less likely to have bad symptoms from it. And it's like anything, if you want to be a better athlete in whatever sport you're in, you're going to have some aches and pains, right? I mean, you get those aches and pains, but your body builds up stronger because of it. So with the COVID vaccine, especially, there are some side effects after getting that vaccine, but you're recovering, your body is getting better, it's getting stronger each time. So please get your vaccinations is what I'm trying to go with. This particular person has now had COVID twice, and they've passed it on to other people, including people in their family who are immunocompromised. So it's ridiculous. Sorry, that is my high horse, and I will now get off of it, and I'll actually talk about what we're going to do today, which is meiosis. All right, so meiosis is different from mitosis, and we all know this, right? Because we've seen the Amoeba Sisters now. They're kind of fun, right? Rather than me, you get to see Pinky and I can't remember what the other character's name is. Pinky and Blinky? I don't know. I know one of them's called Pinky. One of the narrators. So meiosis's job is to take the chromosome number in your cells and to change it. Because what we're going to produce in meiosis is we're going to make, what are sex cells called? Gametes. We're going to make gametes. And we want to make sure that our chromosome number is correct because we're going to have a sperm come together and fuse with an egg. And in doing so, we need to have that chromosome number be half the number of our total chromosomes. So for us, we're going to take that chromosome number and we're going to go from 46 and we're going to go down to 23. Okay. And that's what meiosis does. It takes the number and it goes from 46 chromosomes to 23 chromosomes. So it takes it from a diploid number, 2 sets, down to a haploid. Okay. I know there's something else I was going to say. Oh, I was going to then tell you. So we're making sex cells here. So here's my bad joke. Why is meiosis better than mitosis? Because sex sells. Oh, yeah, bad. I know. There you go. There you go. Okay, there it is. There it is. So the thing about meiosis is it does have the same stage names, but go back up here. There's 2 divisions. There's a meiosis 1 and there's a meiosis 2. So rather than mitosis, which only has 1 division, you're going to have 2 divisions in meiosis. And that's one of the differences. And there are others as well. Earlier in my screencast, I talked about this idea that we had homologous chromosomes. So homologous chromosomes are chromosomes that code for the same traits, but they might not give you the same results. Okay, so they have the same genes on them, but those genes might code for a different outcome, they might have different alleles. Okay, so we used that word allele before, the idea that a gene codes for eye color, but the eye color could be hazel, it could be brown, it could be blue, it could be green. So there's different phenotypes that you could have depending on what the actual code of that gene is. Okay? And those are homologous chromosomes. You get half of your homologous chromosomes from one parent, and you get half of your homologous chromosomes from the other parent. Okay. During meiosis, what we are going to do is we are going to take that homologous chromosome number and we're going to reduce it. So instead of having pairs, 23 pairs of homologous chromosomes, we're just going to have 23 chromosomes. So no pairs, not going to have 46 total. We're only going to have 23 total. Now, to set this up, one of the first things that's going to happen during this process is we're still in the cell cycle. And we go through G1 and we go through S. So when we go through S, we are synthesizing. And what we're going to do is we're going to take our chromosomes and we are going to duplicate them. So up here at the top, I have 2 homologous chromosomes. Notice how they are singles. They don't have anything next to them. Then they're going to go through S. And when they go through S, they're going to get an identical copy of themselves. So now I have 1 and 2. Gesundheit. We call those sister chromatids. Sister chromatids after S should be identical to each other. They're completely the same. Okay? So that 1 blue chromosome is now identical to the other blue chromosome. And the same thing for reds. I've got 2 sisters here. Whee! Okay, so I've got all this genetic information. Over the course of meiosis, we're going to have a couple of different things happen that we're going to spend some time on. The first thing that's going to happen is we have meiosis 1. And during meiosis 1 is where the change in chromosome number occurs. So we're going to reduce the number of chromosomes in humans' case from 46 to 23. So this first picture that I showed you back up at the top here, let me clear that out. This is continued on our next page. Okay, so I've got two sets of sisters. And now in meiosis 1, now I have split them. Okay, I've split them. And in meiosis 2, well, let me say, so up here, up at the top here, I had a total of 46 chromosomes. Okay. Each chromosome has one sister. So how many pieces of DNA do I actually have if I have a duplicate?
46. Wait, like
92. Because each chromosome, so I'm saying I have 46 total chromosomes, but I have a sister for each of those 46. Right? Sorry, I have two sisters for each of those 46. So I actually have 92 pieces of DNA.
But you have 46 chromosomes.
But I have 46 chromosomes. With two, and each chromosome has two sisters.
Right.
Up here, I also had 46 chromosomes, but I had no sisters. So I have no sisters here, side face, and here I have two cis. Okay. I go down here. Now I have 23 in each. Okay. Now I have 23 chromosomes. Do I have sisters?
No.
Look at the picture. Yeah, you do have. See how I've got. Yeah. So I still have 46 pieces of DNA. I have 23 chromosomes, but I have 46 pieces of DNA. We call meiosis 1 the reduction division because it's reducing the number of chromosomes by half. And this is where it gets a little bit funky because it's like, well, wait a minute.
Wait, reducing the chromosomes by 2?
By the chromosome number by half. You still have the sisters. Yeah.
So if they're exactly identical,
Yes.
how do they know which one's the chromosome and which one's the sister?
They don't.
Uh-oh.
They are randomly pulled apart.
That's right.
So they are being shown right here where I think blue in the book is supposed to be from the father and red is from the mother. But they wouldn't have to be pulled apart in this particular way. Remember, we're just looking at one chromosome here. We'd actually have 23 X's in there.
23 of the X's.
Yeah. So remember that we really should be showing 23 X's in here. I'm not going to do 23, but you get the idea. There's lots of them. I'm just showing you one for the sake of simplicity.
So it doubles them, and then it splits them. So it's just doubling and splits.
Yeah.
Okay.
So in meiosis 2, they're going to split again. But we still say the chromosome number is 23.
No sisters.
And we have no sisters. Boom.
Bang.
Okay?
There's two sets of 23, right?
How does that work, though?
So we've got sisters. And here we no longer have.
That's actually free once.
So we have 4 sets of 23 now?
Yeah.
Okay. So before we had 2 sets of 23 with sisters, and now we have 4 sets of 23 with no sisters.
Correct.
Wait, 23 is the chromosomes, right?
Number of chromosomes.
But it'd be like 46 DNA.
Say it again. It would be you have 46 things of DNA here,
Yeah.
but not here.
You would have.
You only have 23.
You only have 23
So
now.
it's just one more step
Yeah,
for
it's actually
mitosis. It's just that mitosis went one
Okay.
more division.
I stopped there.
Okay.
Yeah, very much like that. Yeah.
Not gonna lie.
All right,
I
cool.
was getting it and
All right.
that shit kinda-
Let it kind of sink in there because that's kind of That's why I wanted to be in class, because I knew there was going to be like a moment where you're trying to grasp that, because it's annoying the way that they count it.
Why do they do it like that?
All right, so there's meiosis 1, there's meiosis 2. The very first thing that's going to happen is meiosis 1, right? We come in order. And we're going to have these same stages that we had in mitosis, but now we're going to put a number after it to tell us that we're talking about meiosis rather than mitosis. Okay? You're not going to need to worry about these specifics that are happening in these stages, but there's a couple of things that are really key that I want to talk about. And one of those happens very early on in meiosis 1, which is prophase 1. And that is something called crossing over. Has anyone heard of crossing over before? Maybe from the video, or maybe from earlier days? Okay. Like when you were youngins? Like freshmen? Maybe? I hope. I don't remember anything. Okay, great. All right, so crossing over is huge. It's really important. And it makes things that sexually reproduce, it gives them variation that other species that don't reproduce sexually, the non-sexually reproducing species, it's a disadvantage for them. Okay? Because what we're going to do with crossing over, what we're going to do in this prophase 1 is homologous chromosomes at the very beginning of prophase 1. They're going to line up next to each other. Okay, so this might be chromosome number 1. Let's call him number 1. He's a nice big one. And so this is the, we could say that this is parent 1, and this is parent 2. Okay, these are that organism's chromosomes. And remember, we have one sister, one sister, one sister, one sister. Okay. So in the next figure over here, what are they doing? They're crossing. They're holding hands. Okay, they're holding hands. When they release from each other, so that crossing over is this little bit here. They've crossed over. But when they cross over, they give a little bit of themselves to the other one. Isn't that cute?
Cute wholesome chromosomes.
Exactly. So now this is P1, this is P2 still. But now, parent 1's chromosome, one of the sisters, just one of the sisters, now has some DNA given to them from parent 2. And the same happens for parent 2 chromosome. This chromatid has some of parent 1's DNA. So now those sisters that we said were identical right after S, they're no longer identical. Let me see. Questions on that before I go forward? Maybe. I don't know. Don't know enough to ask a question yet. After interphase, the sister chromatids are held together by proteins called cohesions. Non-sister chromatids are broken at precisely corresponding positions, meaning that when that crossing over and that breaking off of DNA happens, if it's 50 chunks of DNA or 50 pieces of DNA, I shouldn't say pieces. If it's 50 genes that are going to the one parent, it's 50 genes that are going to the other parent. So they get exactly the same amount of genetic information exchanged. So it isn't like one gives more than the other. They give exactly the right amount. This part where it breaks off and it comes back together is called synapsis. And it's going to be reformed. So there are enzymes that will go in there and they will, here's our little homologous chromosomes. They're being held together. The complex is called a synaptonemal complex when they're held together like that. So that picture up above, the one that I showed you earlier, this one, they don't actually look like this. They're actually held one on top of each of the other one. They really look more like that. We just show it that other way because it's easier to visualize. And some of that information between one parent and the other will be exchanged. Okay. Now, next thing that's going to happen is those homologous chromosomes, after they've exchanged information, after they've crossed over, they will line up along the middle of the cell, and they will get pulled apart. And they're going to get pulled apart in anaphase 1. And then the cells will start to reform. And that's meiosis 1. So there's two big things that have happened in meiosis 1. First, there's reduction division. During anaphase, we pulled apart the chromosomes so that now we go from a chromosome number that's 46 to 23. The other thing that we have done is we have created sisters that are no longer identical to each other because of crossing over. So, really important at this point, and it's one of those questions that comes up in all sorts of forms on tests, is between meiosis 1 and meiosis 2, there's no chromosome replication. There's no synthesis. There's no S. Because if there was an S, you would undo everything you just did. In meiosis 1, you took the chromosome number and you went from 46 and you went down to 23. Okay. This is meiosis 1. If we go through synthesis, we're going to bring that number back up again, and we don't want that to happen. Okay. Now, I'm going to talk a little bit more about this whole thing because I'm seeing a lot of confuzzlement on faces. Probably not the best place to put it, but it's well, let me get into different. I don't know if it really matters at this point, but the synapses is just where there's proteins that are holding them together, and that is the synapses. So they're being held together. So when one moves, they both move. And then the crossing over is where they actually touch and exchange information. And this whole thing is called the synaptonemal complex. It's got a funky word for it. You knew that? Is that what you said? All right. So one of the things that I want to talk about now, and I'll come back to it later as well, is actually, maybe I'm not going to talk about that yet because I think there's a better picture than what I can show. Yeah, I'm going to hold off on that. All right, I'm going to hold off on that thought that you don't even know what I'm thinking. All right, so here is a picture of all that happening. In prophase 1, they're touching, they're exchanging information. Each sister now has different DNA from the other parent that it didn't have before. So they are no longer identical. Each chromosome pair, I think it's something like there's 2.5 crossover events per chromosome per division.
2.5.
Yeah, well, on average. It's an average. Yeah. So it isn't just once this happens. It happens usually a couple times. So in the end, the sister chromatins end up being very different at the end of meiosis 1. Okay. Then no cell division, and we're going to go directly into meiosis 2. Meiosis 2 is going to have these same phases, but now we're going to put a 2 underneath it or 2 beside it to show us that we're talking about meiosis 2. And we're not talking about meiosis 1. We're not talking about mitosis. So now we have 23 chromosomes, but what do we still have that we could tear apart? We can split.
The chromosome sisters.
The sisters. It's time for sisters to leave each other. Wah, wah, wah. So now the sisters are going to line up, and they are going to be split apart. But what's being split apart now are the sister chromatids in anaphase 2. So we go through the whole process again.
I think you should put pictures on it.
Pictures would be great, wouldn't they?
Yeah.
Like
Pictures
not
to-
the pictures at the start and then go through the pictures right out.
Um,
I'm kind of lost.
okay, let's.
Uh-oh.
No, okay. This is prophase 2, but it's really small. And now in prophase 2, we're starting off, we have 23. We have 23 sisters. They're going to line up down the middle like they always do in metaphase. But now it's being split apart. One sister's going this way. One sister's going that way. And remember, how many cells did we create in meiosis 1? 2. So here's one of those cells. And there's a second cell.
So that's like the blue. That's one is the blue and one is the red.
No, it's red. Still red.
It could be if, yeah,
But like that's in the early.
in the earlier one, yeah. Yeah. This one, they're now showing it a little bit different. But yes, you were right. So then now how many we're going to have at the very end? These guys are splitting. How many cells am I going to have now? 4. Okay. Each of those cells from meiosis 1 are going to split again. And we're going to end up with each cell now having 23 no sisters. Okay. Each one of those has 23 and no sisters. Marin.
0 sisters.
Yes, that's a good question. So there are what we call germline cells. And those germline cells are the ones that will go through this process. And they will they'll go through this and through mitosis. So germline splits. Let me add a picture in here. So I have a germline cell. And through mitosis, it'll make 2 new cells. One of those cells will just stay a germline cell so that you have a backup of cells. This one, though, will go on and it will go through meiosis. Okay, but you'll always have a cache of cells in reserve, because if you only take one of the ones that you split, you hold the one back as a germline cell, and the other one goes through meiosis. Good question. All right. Comparison between mitosis and meiosis. Mitosis conserves the number of chromosome sets you have. So these are body cells or somatic cells. This is going to take our number, and for 46 to begin with, we're going to be 46 at the end. We're producing identical cells. That's a huge point with mitosis. We want everything to be the same. Meiosis, on the other hand, it's going to reduce that chromosome number by half. So we only have, in case of humans, 23 chromosomes And are they identical to each other? Why not?
Crossing over.
Crossing over is a big one. So because of crossing over and because that DNA gets exchanged between the parental chromosomes, you end up with sisters that are quite different from each other. And remember, that was just one chromosome that we looked at. We weren't looking at 23 pairs. That was just one pair we were looking at. So all those crossing over events are very important. So 3 events that are unique to meiosis and happen all in meiosis 1. That crossing over is one of the events. That's one of the big things. And that is going to make the cells very different from each other. Second one is the way the homologous chromosomes line up. And I'm hoping, no. Let's see, maybe here. Oh, yeah, okay. This one. Homologous chromosome pairs line up at the metaphase plate. And how they line up can be different between each set. So let me go down here to, I'm going to go forward a few slides to this one. So here I have a total of 4 chromosomes. And on the left-hand side, I have both of the blue parents, the blue parental DNA, lining up on one side and the red on the other. I mean, this is obviously very simplified. So I end up with this combination where 2 cells have just one parent, the blue parent's DNA, and these guys have the red parent's DNA. That's not really going to happen. This is meiosis. This is when they line up, when those homologous chromosomes line up. Each pair of homologous chromosomes lines up independently of every other pair. So here, chromosome pair number 1 decided, yeah, I'm going to line up just like I did in possibility number 1. And then the one down there said, no, we're going to line up reverse. So as a result, these guys, oh, I should just go down to the bottom. These guys have a different combination of parental chromosomes because of the way they lined up. And that does not take into account crossing over. So crossing over isn't even shown in this. Okay. Where were we? Were we there? Yeah. Okay. So those things make meiosis unique, and they end up giving you very different gametes. Very different gametes. Okay, so every gamete ends up being unique. This is a summary of all that, kind of broken down versus meiosis versus mitosis. How many DNA replications are there? How many divisions, et cetera, et cetera. So it's a good one to review from. I wanted to just end with this 13.4, which is genetic variation. So one of the things that is a prime source of variation in any species is mutation. So mutation is just a change in DNA sequence. So if the DNA changes in some way, that's a mutation. And that could be good, it could be bad, it could be nothing. Usually it's nothing, but it could be bad. In the case of things like sickle cell anemia, for example, not good. It could be good, and there are some examples of that. There's a antifreeze gene that has mutated. Used to be for, I can't remember for what else. We'll get to that when we get to evolution. But it used to be serve. I think it was a cell respiration function, and now it serves to keep fish from freezing when they're when they're in ice cold water. Okay, so that's a good mutation. But not only do we have those mutations, but we also have this variation that happens during meiosis that happens as well. So because of that crossing over, you end up with unique combinations that are a combination of both parents in unique ways. So what that, I don't know if I can get to this point right now or not. Let me see what this one says. The behavior of chromosomes during meiosis and fertilization are responsible for most of the variation that arises in each generation. So the essential question for this unit was, why do I look like or don't look like my biological siblings or parents, for that matter, either one? And the answer is really meiosis. Because of crossing over, because of the fact, there's a few things. Crossing over is one of them. There's also this independent assortment. So let's just go through them. Independent assortment of chromosomes. Homologous pairs of chromosomes orient randomly at them during metaphase 1. So how they line up, that has to do with what I showed you here. Okay. Each pair, they could line up one way or they could line up the opposite way. Okay, so it's like a 50-50 shot which way they line up. Are they on the left side or are they on the right side? Which side are they on? But that then happens for each of the 23 different chromosome sets. Okay, so imagine the idea that all of the chromosomes lined up exactly the same on one side and got pulled apart. Right? Not very likely that that's going to happen. It's probably going to mix up. That during meiosis it gets mixed up, and that's called independent assortment. Crossing over, we already know that they touch, they hold arms for a minute, and then they snap off and they exchange information. How that happens, completely random that we know of, and different each time. So those crossing over events are completely random too. Then you produce these 4 gametes at the end. Which gamete fertilizes, or which gamete is fertilized, is also random. Right? So which egg is fertilized by which sperm is a random event. Okay? So there's random fertilization, there's crossing over, there's independent assortment. So when you get down to it, the number of combinations possible when chromosomes assort independently into gametes, okay, do they go to this cell or do they go to that cell, is 2 to the n. You will not need to remember this, but that means that there's more than 8 million possible combinations of chromosomes when you're dealing with 46 total. That doesn't take into account crossing over or the fact that which sperm fertilizes which egg is completely random too.
One in like a billion.
Right. So, there are instances where you're like, oh, I saw your doppelganger the other day, right? You saw somebody is like just like you, right?
Yeah.
So it is possible for you to get some similar features, but it's not very probable. It's highly improbable. Okay. Yeah. So. Crossing over. Yeah. All right. So, one more slide and then I'll cut you loose. Meiosis has a huge impact in terms of evolution because it creates this ginormous diversity. Because every time these sperm and egg cells are made, they're different. They're unique. Some of them are going to be good. Some of them are going to be not so good. But it's also going to depend on what situation they're exposed to. Okay? I don't have any clue why I haven't gotten COVID yet. I haven't, I don't think, gotten COVID. Okay? So, In the case of COVID, I might have better genes, maybe some extra resistance in terms of respiratory issues, but in terms of influenza A, not so good, right? So it just depends on what the environment is. If you're exposed to one selective pressure, you may do very well in that environment. You might not do as well in a different environment. Point is that because we have sexual reproduction, we have meiosis, we have this genetic variation because of crossing over, independent assortment, random fertilization. All of that gives us this huge variety to play with. So no matter what the environment throws at the species, if they are sexually reproducing, they should have a better chance of surviving if there's a change in conditions. Organisms that are asexual, not so good. The environment changes, everybody's a clone for the most part. Right? There can be mutations, and that can lead to success, but there's no sexual variation like there is in meiosis. Okay, so that is a definite advantage for species who perform meiosis and are sexually reproducing. Okay, all right, cool. Tomorrow I will hit you with the why you may think you're one thing, but you're actually not, in terms of, like, I like to think I'm Norwegian. My dad was Norwegian, somewhat. But am I actually Norwegian?